Okay, if Dr. Smirnova, if you are ready, we should start on time. Okay, okay.
Sounds good. Yes.
Welcome to our first virtual statistical seminar. Hope everybody had a great summer, or should I say, I wish everybody had a great summer. Mine certainly wasn't the greatest ever. Since this is our first virtual seminar, I would like to say a few quick words very quickly. First of all, please mute yourself when you come in and keep muted throughout the talk. And that's when you are talking to the speaker or asking questions. And once you are done, don't forget to mute yourself again. As you have noticed, the seminar has been changed to 12 to 12:50, which will be used throughout this semester. I believe we can hang around a bit longer after 12:50 if any of you have remaining questions for the speaker. When you ask question after the talk, I would kindly ask you to use the raise hand function in Zoom, and I will call your name, just to avoid several people speaking at the same time. So, okay. So it's my great pleasure to introduce our speaker today, Dr. Smirnova from the Department of Biostatistics at Virginia Commonwealth University. Before she became assistant professor, she got her PhD from UT Dallas in 2014. Her research interests include the development of statistical methods for high-dimensional data, such as microbial metagenomic sequencing data, as well as data collected from wearable devices. Today, she will tell us about her work on physical activity measured by wearable accelerometers in large population national health and nutrition examination studies. If I can ask Dr. Smirnova to share a screen. Well, let's welcome our speaker.
Thank you. So let me share my screen and do you see the presentation?
Yes, I can.
Okay, I'll take away all the faces so they don't interact with the slides. So can you see the screen well and hear me well?
Yes, yes.
Okay, perfect. So first of all, thank you very much for inviting me to present that virtual seminar. And it was a pleasure to meet with the faculty members earlier and discuss our research. Now I'm going to discuss the part of my research program that is related to high-dimensional regression with survey design. And specifically, I will concentrate on the association of physical activity and mortality in a National Health Examination Survey, or short NHANES. First of all, the motivating problem for this project is predicting 5-year mortality using physical activity data. And here in this figure, I have a set of six participants from the National Health Examination Study. And those participants were chosen at random from the whole data set. On the x-axis, we have time during the day, and the data is collected at minute level. So here, each trajectory corresponds to the participant's physical activity throughout the day. And one trajectory corresponds to one participant. So as we can see, we have different activity profiles, slightly different activity profiles for the people. However, what we are really fundamentally interested in is looking at these blue curves for those participants who died after 5 years from participating in the survey, from those in red who were alive. And our goal is to differentiate those curves and associate them with the mortality outcomes. So fundamentally, there are a number of methods that allow us to answer those questions. And while this is physical activity data, this is data that is collected according to special design, which is essentially not selecting participants entirely at random. And because of this not entirely at random selection, it poses a number of methodological problems that come in analysis of such data. And throughout this presentation, I will discuss it in more details and provide some solutions that we came up with in order to do this analysis. So when I mean that participants are not selected entirely at random, it's the selection that the NHANES study, which is conducted for the Centers for Disease Control, is imposing on selecting the specific individuals. Specifically, when when the individuals are selected, the geographically related counties are selected. Then from those counties, segments, households, and finally individuals. Also, in this study, some individuals are oversampled. For example, children are oversampled, as well as older adults. Also, the design imposes the specifics of the geographic regions where the participants are selected. And this is done for the purposes of study being more cost effective, in order to select as much population sample as possible, at the same time accounting for the fact that we want to make that sample as representative of the whole US population as we can. So as a result, what it leads to is the fact that we don't have the technically IID sample in our data. And this kind of clustering design occurs not only in the national health examination studies, but also in a number of other survey studies, as well as sometimes implicitly there is a special preference of the geographic region where the participants are regions where participants are selected, for example, in multi-site studies. And that imposes some clustering relationship that is coming in this data. So the fundamental question that we are interested in is predicting health outcomes, here denoted as YI. In our case, this is the binary mortality status, dead or alive in 5 years, using participants' health characteristics, which are BMI, age, and other health status, as well as the functional signal, ZI, which is physical activity in our case, over the entire time domain, which is in our situation is the time throughout the day. Such questions could be answered using the scalar function, scalar response, and functional predictor regression, where in this model, the left-hand side of the equation is the link function for the exponential family of regressions. And the first term is the familiar regression term with p covariates, which correspond to our participant health characteristics. And the second term is the functional part, which takes into account the activity throughout the day that is taken into account throughout the whole time domain. There are a number of methods that are developed for fitting such models. And here are some references that develop the model fit. For the purposes of these models, essentially what we are thinking of here is that the participants are independent. And in order to fit these models, there are a number of ways of doing this. But one of the simplest ones is to project gamma of t, which are the functional regression coefficients, onto the basis system, phi, which is denoted here as phi k. And then the integral part of the regression equation can be represented in terms of this basis system, where theta k is the coefficient of basis representation that corresponds to each term, each basis element phi k, and rearranging the terms in the integral, we can look into this, describe the functional part as the coefficients wik, which are the finite coefficients. Using this representation, it allows us to use the least squares fit in order to estimate both the beta, which are the traditional coefficients in regression, and gamma, which are the functional coefficients, which are now discretized according and correspond to each basis element. The unpenalized fit is looking for solutions, beta hat and gamma hat, that will minimize the least squares. However, because the gamma here are the functional regression coefficients, actually the penalized fit is used, where P here is the penalty function for this functional coefficients gamma K that impose the smoothness in those coefficients. And lambda is the tuning parameter that controls the smoothness. The higher values of lambda would inform greater smoothness. So the goal is to extend those methods of functional regression to our specific design of complex-based survey sampling. And finally, answer our questions about a relationship between physical activity and mortality outcomes. So what is this design-based data? So the way that this sample is collected, for example, let's take a simple example first. We are collecting the sample of n individuals from the population of size capital N, and this is the finite population size. Each individual is, if it is simple random sampling, then each individual has an equal chance of being selected, and the selection probability is the number of participants divided by the total population size. As a result, if we invert that selection probability, we know that each individual represents W individuals in the population, and this is the weight that each individual has in the sample. When we do design-based sampling, where we are concentrating on special groups of interest, for example, racial, social demographic, or even geographic areas that are sampled, we no longer have this simple random sampling. And as a result, the chances of an individual being selected vary across individuals. This can be taken into account by assigning weights, WI, to each participant, so that these weights add up to the total population size. And again, these weights will tell us how many individuals the participant represents in the population. Also, again, because there are specific groups of interest, geographic areas, and so on, this imposes an implicit clustering for the place where the participants were selected. Now, how it comes into play in methodological challenges. So there are a number of challenges that occur because of this sampling design in inference. First of all, we have to realize that participants are no longer sampled independently. And we need to take into account the participant weights in order to make the inference representative of the population. Also, the standard error estimation is again affected by design. There are, of course, a number of solutions that already this problem has been known for a while, and there are a number of methods that already exist for this complex survey weighted regression, and there's implementation for linear and generalized linear regression, and it's available in R package survey. However, there are no methods for functional data yet, and this is what we are interested in doing in our research. The current solution that we have implemented at first would be to describe the functional data as day-level summaries. For example, if we are thinking of the entire time domain from for the time during the day, the data is collected at minute level. And for every minute, we have a count, which is corresponding to the activity count of that participant during this time interval. So we can add up all these activity counts throughout the time domain, and describe it as the total activity count, which would represent the day level summary. There are also a number of other day level summaries that are attempting to capture the high-dimensional nature, of the data, and the fact that those physical activity trajectories are not uniform across, throughout the day. For example, one of the popular summaries would be to look at the time the person spent in moderate to vigorous physical activity, which would be examples of running, fast walking. And another way of representing this is to look at the functional principal component scores and make the data-driven summaries. All of those are solutions that we implemented first, and I will first show some results that that we have using the day level summaries, and then we'll move on to the part on functional prediction. Before I go into details about the actual model fitting and methodology, I'd like to point out that this is the data that is high dimensional data collected throughout the entire day. So every time domain is about 1440 minutes, and also collected throughout multiple individuals, actually thousands of individuals for this population size. The data is publicly available. However, there are a number of practical challenges actually working with this data. Although it's available, there is no clear no clear reference on how to begin working with NHANES. The documentation on this data set, which is the study conducted by the Centers for Disease Control, is available on the CDC website, but it's a vast documentation that actually has multiple, many files for different covariates, lab measurements, mortality, which is linking those files to the registers, and so that we can see even a while after the participant has taken the survey, we know later on the mortality outcomes. Accelerometry is a separate set of files that is also, all of those together need to be linked, and it requires non-trivial amount of background information going through these details of NHANES, and also knowledge of data linking and processing. In addition to that, one needs to understand the details of working with accelerometry. First of all, this is the data that measured for 7 consecutive days for each participant. It also requires some sort of quality control. For example, wear time. Those participants are regular people from the US population, and although they were instructed to wear the accelerometer throughout the day, except for taking it during the swimming or bathing, it does not necessarily mean that every single person actually was compliant. As a result, we need to be careful about the time that during the day, so that it's actually enough readings from the accelerometer in order to say that this is a valid wear day. Finally, we either have to work with this as functional data or develop the effective summaries in order to catch high dimensional features. The solution to this data organization problem is now available in the R package, rnhanesdata, which is being developed by Andrew Leroux, who is now joined as an assistant professor at the University of Colorado. And overall, the project was led as a part of the Wearable and Implantable group at Johns Hopkins. And we have a publication that describes the details of how we organize this data and some of the analysis that one can do with activity data if they are interested getting into this area of research. Also, we have that package rnhanesdata that is available on developers' website at developers' GitHub website. And in addition to that, we also created package vignettes, which allow one to in a very short time frame to get familiar with this data. The whole project took about about 6 months in order to put this package together, make the vignettes, make it publicly available, and so on. So I'm saying this in the sense that this is a very part of the things that sometimes we do not realize how much time actually goes into putting these large data sets together. So the first things that we've done is we looked at the mortality prediction using physical activity daily summaries. And this was using the 2 waves of NHANES. And we applied a number of exclusion criteria. For example, those who refused to answer medical condition questions were removed. Also, because we were interested in natural mortality, we removed the people who died of accident or unknown causes of death, and made sure that we have enough accelerometer wear time, at least 10 hours. And for the first look at the association, we used the activity derived summaries, which are day level summaries, and we examined a number of them together with the history of chronic diseases, alcohol consumption, and other physiological measurements. So here is the short snapshot of the relationship between this physical activity and mortality using univariate logistic regression. So this is the simplest analysis possible that we can do in order to have a gain understanding in how each of this physical activity and other traditional covariates such as age, mobility problem, drinking, smoking relate to outcomes. The univariate logistic regressions essentially look into each of the variable at a time as the predictor and see what its corresponding predictive power measured by AUC. In this table, we have it ranked by AUC criteria. And what was striking is the fact that physical activity variables are essentially the ones that are colored in red, are among the top most predictive variables of mortality. So that gives us the idea of how important this studying physical activity is in health research. We also looked at the subset of these predictors in order to understand, in order to select the best subset of predictors and develop the model for mortality prediction using the simple daily summary as the first step. We looked at the criteria according to the cross-validated AUC, which is this red line here. And the stopping rule was the point when AUC, cross-validated AUC starts decreasing, which corresponds to this red dot. So looking down, we have the best subset model, which consists of 3 different activity measured summaries, total activity count, and 2 other activity-derived measures. And together with the age, mobility problem, drinking, and smoking. So what is striking is that actually 3 different different types of activity-derived measures are used in this best subset model, which gives us a suggestion that probably one single summary is not enough to describe the whole complexity of this functional trajectory. and probably we need more other methods in order to capture this high dimensionality of the data. Also, we looked at the correlation between all these activity-derived measures, and although they are quite highly correlated, the correlation is not perfect. It's the correlation values are not like in the range of 0.8. They are mostly in the range of 0.7, and for some of the activity-derived measures, they are actually quite weak. Especially this, the one that's very interesting is this feature that is derived from the functional principal component scores, and it's extremely weakly correlated with all other activity measures. However, it plays a very important part in the mortality prediction. So now I would like to move on to the part when we actually are trying to develop the techniques for taking into account the full functional profile and use the multi-stage sampling design in order to do the functional regression. So the data structure for this enhanced multi-stage design is that we have sampling strata, and there are 30 of them in this case, and 2 principal sampling units per stratum, which is the most common design in such survey models. What it leads to is that we have a total of 60 clusters, where we can, the way that we think of those clusters is that there is some relationship between the individuals in the way they were chosen and some similarity by geographic location across participants in those clusters. So that leads to the data for each participant with corresponding subject weights that are non-zero. So the subject weights again represent the number of people an individual person, I, from strata H, and PSU alpha represents in the population. Also, it's important to notice that there are a large number of participants in the cluster. But when we do subset for mortality selection, we apply strict exclusion criteria, and the number goes down in our subset. Also, the participants under 18, which we do not use in our analysis because we restricted to only those above 50, but those over 60 years old are oversampled, which means a large part of our population size for the analysis was oversampled, as well as Mexican Americans, African Americans, and low-income white Americans. This again gives an idea that taking into account that survey-weighted design is important in order to make the inference representative of the population. So I will start giving the idea of how we fit those models using the survey-weighted regression, which is looking at the traditional regression without taking into account the functional covariates yet. So we have the observations y for each participant and the covariates such as BMI, age, from H strata, alpha H PSUs, and there are a number of individuals in the in this combination of strata and PSU, which I call cluster. There are corresponding weights that are associated to each participant, and we can estimate the coefficients for regression using weighted least squares. So the way that the weighted least squares will do is they will essentially take into account those participant weights in the minimization problem when we are looking at looking for the coefficients of regression. So the weighted least squares will give us the correct design-based estimate for the population regression parameters beta. However, when we are thinking of these weighted least squares, it assumes that the individuals are coming from the independent sample. And this is not the case in our situation. As a result, we will have biased estimates for standard errors if we use traditional linear regression models, such as those that are implemented in R function LM. But what's important to note is that if we feed the same LM function with the weights corresponding to participant weight, then we will get the correct design-based estimate for the parameters. And this is what we use in the functional approach. So essentially, we build on that technique from the weighted least squares and use the previously developed methods for functional regression in order to estimate the coefficients for regression and the functional regression gamma using the packages that are implemented in R. So essentially those models could be found via GAM function in MGCV package in R, which takes into account the participant weights. So essentially what we are doing is the smoothed weighted least squares. And the details of implementation and also some details about fitting this class of models are available in several resources. This way, we get the functional regression coefficients gamma, and we can actually analyze this full functional profile. The question that remains is how we find the standard errors for this complex survey design. The first approach that we took is using the principle of the complex survey weighted literature, and these principles essentially are implementing resampling techniques such as Jackknife or Bootstrap, specifically for the complex survey design. So we use the Jackknife for resampling clusters, but what's important is that if we are thinking of the traditional Jackknife for independent sample, at every time in Jackknife, we are dropping one individual eye. In complex survey Jackknife, we are looking at the estimates without the entire cluster, which is the combination of this strata and PSU. Also, the version of Bootstrap for the complex survey design is balanced replicated design, which essentially samples one of the PSU from each strata. To give a better idea of how this complex survey Jackknife works, we are interested in obtaining dataset estimates for the functional regression coefficients. And what we do is we use data without one PSU per each strata. So here in the first dataset, on the x-axis we have different strata, and the y-axis corresponds to PSU. The blue rectangle here is the PSU1 from strata 29 that we do not use in the first data set. So essentially we obtain the estimates using the rest of the PSU and strata. And this way we get the first Jackknife estimate for the functional regression coefficients. At the second step, we do not use the second PSU from strata, and again get the second estimator for the Jackknife coefficients. As a result, we do this for a total of 60 strata, 60 clusters, which is combination of this PSU and strata. So then we can use the naive Jackknife variance estimator, which looks at the deviation from those Jackknife estimates without each given cluster from the estimate that is used for the full data set, which is represented here as gamma. And again, because those are functional estimates, we smooth them by the this basis system. Well, one thing that is really important to note here is the fact that even though we might have a large number of participants within each cluster, for example, the full NHANES design has 500 to 900 participants within each cluster. In our case, because we had a subset of participants, we might have maybe 90 or 100 participants, but still, it's a larger number of participants, and yet, according to this survey design, we are the number of degrees of freedom are limited by the design, so which is the strata and PSU combination. The final estimates for the variance for functional coefficients can be found as the kth element, diagonal element of this variance covariance matrix. And this is an example of how this Jackknife performs for different data sets. The blue line here is the estimate for the functional regression coefficients for the full data set when we use all clusters and all strata. Each dashed line corresponds to an individual estimate for, again, regression coefficients without one PSU from one strata. The way that we interpret this functional coefficient line is, first of all, let's look at the sign. We have for most of the times during the day, the functional regression coefficients are negative, except for times around before 12 a.m. What it means is that there is an inverse relationship between the physical activity and mortality outcomes. So essentially, higher volumes of physical activity during the day are decreasing mortality risks. And that makes sense because we know that physical activity is highly indicative of our health status. Those who are physically active tend to have been shown to have longer and healthier lives. So for different times of the day, we have different estimates of the regression coefficients, which give us an idea of the times when higher levels of physical activity matter the most. For example, those times during the day, this is when the subjects most likely differ in their physical activity profiles, and that's when it matters, and the coefficients are for regression are much smaller. And what's interesting also is the time during night. So those who are slightly more active during the night times, we do not have explanation why, but maybe because they have some sleep problems or something else. I do not want to be over conclusive, but it seems that there's some relationship that activity during the night is not as healthy. One thing that I forgot to mention is the fact that while most of these Jackknife estimates are somewhat close to the estimate for the full data, there are deviations in several cases. For example, in this one and this line, they seem to be very drastically, much drastically different than the rest of the estimates, which poses a problem of whether this approach is appropriate for this, always appropriate for this scenario. In order to understand what is the reason, we looked at individual activity profiles, and here we have a summary of individuals from, unfortunately Zoom does not show it, but it's one of the strata which is this geographic locations. So the first PSU, the top panel corresponds to those who were alive in 5 years, and the bottom to those who were dead. And essentially what we do is we, at each Jackknife data set, we drop this, all of these subjects. But we can see that activity profiles, especially for those who were dead in 5 years, differ quite drastically across the PSUs. And it means that it tends to ask the question whether, again, this clustering, this approach of dropping an entire cluster of so many individuals is appropriate. Similar situation remains in other strata, where the physical activity profiles are again vastly different. Part of the problem is because we do not have as many events. There are not as many people who were dead in 5 years as opposed to those who were alive. And this is the part where we still have open questions. However, I thought that it would be still interesting and useful to show to you what are the open questions that still remain in this area. We also tried other ways for resampling and standard error estimate. Another approach which is alternative to Jackknife is the balanced replicated design, where we remove we select one PSU from each strata. So here in this example, we have on the x-axis strata, and the y-axis are individual replicates. Color corresponds to the PSU that is selected from that strata. So every time we select one of the two PSUs from strata. So for example, in the first replicate, we select white, PSU2. In the second PSU1. In strata 33, PSU2, and so on. So that leads to only half of data that is actually used in each replication. What we found is that using this method, there are about 10% of replication that lead to large deviations from the estimates using all data. And that is something that we think is very problematic because it's a very large percent, and the deviations could be quite drastic. And we are investigating further the reasons for this, as well as other approaches to the estimation for standard error of these coefficients. One of the other approaches would be to use mixed effects modeling, where we treat cluster as a random effect to account for the correlation structure within this cluster of individuals. We could do it with R. However, if we actually do it with the current implementation, we run into convergence problems because of the interconvergence problems, which we have not resolved yet. Now, I'd like to summarize the part of what we have achieved and what we still have remaining as open questions. First of all, we created this great tool, rnhanesdata, that allows anyone to with our knowledge to very quickly get started to working with accelerometry. The vignettes are very user-friendly, and they go over all the code lines and corresponding description for how we select a subset and how we link the covariates. We also have shown that physical activity is highly predictive of mortality outcomes. And this was done using daily summaries. We confirmed that what I have shown previously was the 5-year mortality outcome. But we also confirmed that the high role of physical activity in 1, 2, 3, 4, 5-year prediction models, where we look at an individual prediction horizon and evaluate the role of physical activity. Again, we find physical activity to be consistently highly predictive of mortality outcomes. Similar results were confirmed in another data set, UK Biobank study, which also uses accelerometry, but it is doing it in the UK population, and also on the larger data set, and with a different accelerometry device. But a similar result, the inferential results again hold the same. Finally, now we are able to find the coefficient estimates for the functional regression in the complex survey models, which is a big step forward. However, what remains is still the open questions of how do we improve our estimation for the standard errors of those coefficients. Some of the things that we need to take into account is proper adjustment of participant weights, because every time that we do these resampling techniques, we drop a number of participants, and we need to rethink the way how weights are adjusted. Also, we are investigating whether the assumption of similarity for activity across clusters is actually reasonable, and whether we can find other methods to adjust for correlation structure without actually using this very heavy cluster assumption. And finally, we need the simulation studies that will properly mimic the this survey sampling design. So those are some of the references for the for fitting the complex survey, for fitting functional regression models, for those who are interested. And I think it, we can stop here, and I would take the questions.
Okay, great. Thanks for the interesting, great talk. Do we have any question for Dr. Smirnova?
Can I ask a question?
Of course.
Oh, yeah, of course.
Yeah, hi. So I was curious with the Jackknife, it's a pretty computationally expensive procedure. So in your case, when you're doing this with all the strata for the NHANES data, how long does it take?
Actually, it doesn't take that long, because it's only 60 clusters.
Oh, right.
And
it's not that
that's
many.
it. Exactly. That's the thing that is that happens with the way that we are thinking of Bootstrap, or we are thinking of the Jackknife, any replication procedure. Usually it's an individual that is randomly sampled, or in Jackknife not used. But in complex survey weighted models, traditionally it's the whole cluster of individuals that is not used. And because we have that design with 60 clusters, it's only 60 replicates.
Thank you.
Any other questions?
I have one. Ekaterina, you use enhanced data, and enhanced data are sort of snapshots from year to year. They are not longitudinal. Yes. How do you get the information about status like that?
It's linked, the data, when the survey is collected, that's cross-sectional. But then the participant is linked to the survey, to the mortality registers. I think they use either Social Security or other information. So at some point, they of course do not release it publicly, but at some point they release the mortality outcomes.
Mm-hmm. Okay. Thank you.
I have a quick question.
Sure.
Hello, yes. How serious is the non-response problem in this particular survey?
I am not aware because this is the survey conducted by the Centers of Disease Control. I am not 100. I cannot really answer that question, but I assume there is some non-response rate.
You mentioned that some people refuse to answer questions, right?
Yes, unfortunately, this is something that I have not personally checked in terms of what exactly the rate. I cannot say on top of my head. We remove those participants, but it's of course quite practical that not every person would refuse answer the questions.
Thank you.
I have another question regarding the enhanced data set. So the first question is, so besides the time series of step counts, do they also have the GPS recordings?
I don't think so.
Okay. And also the second one, so I know like you're using those physical activity metrics, like moderate to vigorous physical activity. So are those metrics like released from this data set, or you're actually using, or your R packages have functions to pre-process from the raw accelerometer data?
This package that I mentioned, it has a description of how we calculate every single one of the activity-derived measures. I did not go over them in great detail in this particular presentation, but the vignette goes into detail of what each of them means, gives references to how they were developed, and also shows how to calculate them.
Okay.
In terms of the moderate to vigorous physical activity, it's actually quite simple. It's a threshold for step counts. So I think it was 2,200 counts, if I remember correctly. So anything above that is considered as moderate.
Yeah. So it seemed like your package can actually take some of the raw, say, measurements from accelerometer and actually give us the pre-processed results for those different metrics, right?
Yes.
Okay, that's great. Yeah, we will check it out. Thank you.
Okay, any other questions for the speaker? Okay, if there's no other question, well, this is a very good talk. Thank you again, Dr. Smirnova. And thanks to everyone for joining today. And we're gonna meet again next Friday. Thank you very much.
Thank you.